Good morning, students. In last videos, we discussed monohybrid and dihybrid crosses. And these observation led Mendel to formulate the three laws of heredity or principles of heredity. The first law is principle or law of dominance. In a hybrid or heterozygous individual, two dissimilar unit and factors are present. We know as these are capital T and small t. Out of these genes or factors, only one is able to express itself and it prevents the expression of the others. Then this is called the dominant gene, this one, and the second one, this which do not express itself, this is called the recessive one. Okay, then according to the first law or the law of dominance, when the parents with pure contrasting traits are crossed together, only one of the trait appears in the next generation. The hybrid offspring will exhibit only the dominant trait in the phenotype. This is the first law of dominance. These two, first and second, these are derived from the monohybrid cross. Right now, see the second. Second is the principle or law of segregation. The two unit factors of a character which remains together in an individual do not get mixed and keep their distinct identity. They separate or segregate during gamete formation so that each gamete receive only one factor for each character and is always pure. Right? And this is called principle of purity of gametes. Now third is this one, third one. This is derived from the dihybrid cross. According to this, the law of independent, this is called the law of independent assortment. Law of independent assortment, right? Uh, this principle states that the unit factor of each character is assorted or distributed into the gamete independently, and gets randomly rearranged in the offspring. For example, in Mendel dihybrid cross, the offspring of F1 generation, when these are self-crossed, R and Y, then when these are self-crossed, at that time, they produce four types of the offspring. Two types were similar to the parent, while the remaining two types had the combination of the traits. This became possible because the unit factor of two characters assorted independent to each other. Right? Then these are the three laws of genetics, which are the conclusion of the experiments performed by the Mendel. Now next topic is sex determination. Okay, what is sex determination? Then what will be the sex of the baby to be born? Is called how it is decided. This is called the sex determination, right? Then simply we can say phenomenon or of decision or determination of the sex of an offspring is called sex determination. Okay, then what are the factors which are responsible for the sex determination? And these are mainly two factors. One, environmental factor, and second is the genetic factor. In some animals, the temperature at which the egg fertilized are kept beside the this temperature is responsible for the gender of the newborn baby. For example, in the turtle, would be baby will be male or female. It depends totally on the temperature. Now next is the genetic factor. Second is the genetic factor. In some animal like the humans, gender or individual is determined. It will be the male or female. It is determined by the pair of the chromosome, which are called the sex chromosome, right? In our body, we know if we are talking about the humans, there are 23 pairs of the chromosome. In total of we can say 46 chromosome are present in a cell. In out of these 46, or we can say 23 pairs, 22 pairs, these are called autosomes. Autosomes. These are called autosomes. And one pair that is called, which is responsible for the sex determination, that is called the sex chromosome. What that is called the sex chromosome. As this is also written in it. Then if the combination of this last pair is the XX, then if both of the chromosome in this pair are of the same type, XX, then the would be this is the combination for the female. If this combination is present, then the baby will be female. And if the combination is X and Y, both are of different type, then it will be a male child. This is a combination of a male. Now see, with the help of this, and one more thing which I want to tell you. When I told you this is a combination of female, this condition is called homologous. Homo means same. Homo means same. If both are of same type, then this condition is called homologous. And if both are of different type, X and Y, then this condition is called heterologous. Right? The shape and size of the both the chromosome will differ. Okay. Now in human females, they have two X chromosome, or we can say two XX sex chromosome. Both the number of the sex chromosome are similar, are also called homomorphic, homologous or homomorphic. Or in male, it is called heterologous or heteromorphic, right? Okay, now we will discuss it with the help of this diagram. Now see, male parent, and this is a female parent. In the male parents, in the testes, sperms are formed. These sperms in females in the ovary, ova are formed, which are called the ovas are formed. Sorry, sorry. Now listen again. In female, ovas are formed having the X chromosome. Two type if all the ova which are forming here, they all are of similar kind, but the sperms are of two type. One having the X chromosome, second having the Y chromosome. Right? Okay. When now see, when with the ova sperm having the X chromosome fuses, then the combination it becomes XX. It means would be baby will be female. If Y this fuses with the X, then the combination is XY. Then we can say 50-50% chances of being male and female baby. Now this is called the sex determination. And if we talk about the factors, environmental factor, and I was giving you the example of the turtle, I want to add one more line in it. In the turtle, if the temperature at the time of fertilization is 30 to 35 degrees Celsius, then in this case females are produced. And if the temperature is 23 to approximately 28 degrees Celsius, then the males are produced. Sorry, males are produced. Right? Okay. Thanks for watching me.